Hello everyone, it's Karen. Today, I'm going to teach you all about animal cloning. But first, let's get started with why I choose this topic. I choose this topic because I learned that now our scientists have the ability to clone humans, and I want to find out all about it. In this video, we'll be talking about the mechanism of animal cloning, the disadvantages and advantages of animal cloning, and of course, the ethical issues that rise. So now, let us reveal all the secrets of animal cloning.
The headline on the other major news story today, to which we intend to devote some time, is very simple. Hello, Dolly.
This is February in 1997. This first cloned sheep, Dolly, has created waves of future shock around the world. She's considered the first living clone of an adult mammal. What is actually animal cloning? Cloning is when genetically identical copies of living organisms are produced. So this means that this sheep, Dolly, has the same genetic makeup as the original sheep. And now, I'll explain the mechanism of animal cloning. We'll remove a mature somatic cell, such as skin cell, from an animal that they wish to copy. Second, we'll transfer the DNA of the donor animal into an unfertilized egg cell, which means that this egg cell has the nucleus removed. I will further explain on how they remove the DNA of the somatic cell. Then, the egg is allowed to develop into early stage embryo. Then, this embryo is implanted into the womb of the adult female animal, which is called the surrogate mother. Later on, this adult female gives birth to an animal that has the same genetic makeup as the animal that donated the somatic cell, which we call the mate animal the clone. And this is the whole process of animal cloning. So now, back to our PowerPoint slides. This is a much better picture of animal cloning. And still, the first step is taking a donor cell from the original sheep, and then using electric shock to combine the donor cell nucleus with the empty egg cell to form the fused cell. Later on, the fused cell will divide and develop into an embryo. Then the embryo is placed into the womb of the surrogate mother, and eventually, the cloned lamb will be born. So now, I'll be showing the steps of transferring DNA. So this peptide is poking through the membrane in order to get the DNA out. As you can see in the peptide, it is aligned in the nucleus, which has all the chromosomes lined up. And now we have this empty egg cell with its nucleus removed. And now I'm going to show you a different step, which is injecting DNA into the empty egg cell. So here's our empty egg cell. And our peptide carrying the nucleus of the somatic cell will poke through the membrane and inject inside. And the peptides actually contain electric current within. So for me, this is kind of like poking through a water balloon. We need to be careful and use this electric charged peptide, so our membrane will not break and our nucleus will not burst out. Now let's look at the difference between gene cloning and animal cloning. So the first step of gene cloning is to cut the plasmid DNA and the cell's DNA with restriction enzyme. So now, you have them combined by using DNA ligase. Keep in mind that gene cloning is to clone the plasmid DNA. But for animal cloning, we clone their egg cell and later on allow it to develop into embryo and into clone. Then, we'll place this new combined plasmid back into the bacterial cell. And as this bacterial cell replicates, it is also the clone of cells. Now, let's talk about the reasons why people want animal cloning. First, look at the graph. This graph is a species extinction since 1800s. As you can see, the rate of the species that extinct after 1920s rise crazily. And this is when we need animal cloning. Once the scientists have preserved the extinct animals' DNA, we can clone them and bring them back to life. The second advantage is that it offers benefit for medical research. It provides more genetically identical organisms so that the result from the experiment will be more accurate. Also, it increases the supply of meat and milk in the market. And of course, this is such a huge breakthrough of genetic engineering. So now, let's talk about the disadvantages of animal cloning. Animal cloning reduces genetic diversity because of creation of identical individuals. And second, the surrogate mothers have high death rate because of the painful hormone treatment and abnormal fetal development that threatens their life. They often caught the disease called hydrops, which is a swelling of fluid in fetus and making the fetus look like it's about to burst. Third, it's a high mortality rate of cloned organisms. Look at the graph on the right. We can see that the dark blue is a normal organism. The lighter blue is a cloned organism. And the disease that's caught by the cloned organism is so much higher than the normal organism. The cloned organism often cannot live more than few weeks because of their high death rate. So after introducing so many advantages of animal cloning, this is science fiction technology had now been accepted by all the people? I believe not. More than 64% of Americans think that cloning is morally wrong, and more than 63% of them will refuse to buy cloned food even though if they are labeled as safe. As the technology of animal cloning rises, this conflict also continues. Many feel that cloning is not natural and is an action of man playing God. Thank you all for watching this video, and I hope you enjoyed this as much as I enjoyed doing this project. And I hope all of you will have better understanding about animal cloning. Thank you.